Namaskar, dear students. Welcome to our channel Bio by Gulab Sir. As a part of series of lectures, we had already started Animal Kingdom with its general characteristic, which is supposed to be considered to classify and study the various characteristics of different phyla. Very first, that either that embryo, which is supposed to be base for development of that phylum, are diploblastic acylomate, triploblastic acylomate, triploblastic pseudocoelomate and triploblastic coelomate. Here you have confusion. What is it exactly? कितने सारे स्टूडेंट हैं वो बहुत कंफ्यूज रहते हैं कि ये टर्म है क्या, ये किसके बारे में है, ये क्या बोला जाता है. पोरीफेरा डिप्लोब्लास्टिक acylomate, सिलेंट्रेटा डिप्लोब्लास्टिक acylomate, चिनोफोरा डिप्लोब्लास्टिक acylomate, प्लैटिहेलमिन्थस ट्रिप्लोब्लास्टिक acylomate. आखिर है क्या? सो बहुत सारे स्टूडेंट को कंफ्यूजन रहता है. मैं वो क्लियर करना चाहता हूँ. मुझे आशा है कि आपको आज के बाद इसके बारे में कोई समस्या नहीं रहेगी. दूसरा, इसके ऊपर नीट में बहुत क्वेश्चंस आते हैं ऑन द बेसिस ऑफ एप्लीकेशन ऑफ नॉलेज, तो इसलिए क्लियर होना बहुत जरूरी है. इसी को ध्यान में रखते हुए मैंने रिपीट एक सेपरेटली वीडियो बनाया है, जो ऑलरेडी इसके बारे में एक्सप्लेन देने वाला वीडियो हमने बनाया है, लेकिन आज आपको एकदम क्लियर हो जाएगा. तो ये वीडियो देखना ना भूलिए. आपको अवश्य वीडियो देखना चाहिए, पूरा वीडियो देखना चाहिए, ताकि आपका एक बेस्ट जनरल कैरेक्टरिस्टिक बिलोंग टू ऑल फाइलम विल बी क्लियर. And moreover, in evolutionary history you will enjoy this knowledge. So let us start. See, here I did mention that embryology development of stages of embryo. Here first stage is zygote, and that too with zona pellucida. Zona pellucida is a outermost layer that has to be understood, because you must know what is this. This is a zona pellucida. Now this is protective covering over the zygote, and this is continue up to this stage. Okay, now zygote develop into two cell stages, that is what undergo mitotic division, and this is a successive different stages of mitotic division without growth. It's called cleavage. This is what embryo development begins with. Zygote undergo mitosis to form 2 cell stage, 4 cell stage, 8 cell stage, 16 cell stage. This stage is called morula. You are seeing here the size of that zygote is as similar as size of this morula. What is increased? That is nuclear material is increased, cytoplasmic material is shared. So nuclear material is increased. That means mitosis without growth is known as cleavage. That is first point is clear to you. Question can be asked neat. What is cleavage? Where does it occur? Or cleavage is करके आपको question पूछा जा सकता है. So see, up to this stage, morula stage, and then morula, each cell is called as blastomer. This each cell present here is called blastomer, and that has to be understood. What is the importance of this blastomer to form this blastula? This stage is called blastula. Here I do write. So please remember here this I am writing the name of that stage is blastula. Arrangement of this blastomer into two parts. One outermost covering called trophoblast, and this inner mass is called embryo mass, from which all these different stages of embryo are developed. Now see, up to this stage, right from porifera to human being, all stages are common. That is called ontogeny of recapitulation. Again, another neat question can be asked. I am writing here this word so that you need not to remember them. Ontogeny of recapitulation. What does it mean? Some of the stages are repeated in the lifespan of every group of organism, although they belong to previous one or advanced one, but they have some of the stages common. That is called as ontogeny of recapitulation, and this phenomenon was given by Hans Haeckel. That also please remember, because question can be asked in it. Now see, this inner cell mass is responsible for these stages. What you supposed to be understand. See, diploblastic acylomate, what does it mean? These embryo mass cells are arranged in two layers in the next stage, endoderm and ectoderm. Because of two layers, ecto and endo, this is diploblastic. Acylomate, it is because of there is no mesoderm, no cavity. So if there is a mesoderm and cavity, it is called as coelomate. Otherwise, it is not called as a coelomate. This area is free, and here that is liquid, non-cellular material is called mesoglia. Therefore, porifera, cilindrata and ctenophore, they are diploblastic acylomate with these two layers. Now next stage, from same embryo mass, these three layers are developed as advance in evolution. That is ectoderm, mesoderm and endoderm. In this three layer, in mesoderm there is no gap, and therefore it is triploblastic because of three layer, but no gap, no cavity, therefore acylomate. So here diploblastic acylomate. Here this is triploblastic coelomate. Now next advanced stage from which it is evolved. So here you can see again this embryo mass develop into ectoderm, mesoderm and endoderm. In this case, in mesoderm you can see fragmented cavity. This is something advanced. That is what evolved from this one. So in that advance, this is triploblastic pseudocoelomate found in Aschelminthus, nematohelminthus. So in the series of this evolution, triploblastic coelomate, now this is in advanced triploblastic pseudocoelomate, where that organ level of organization is started. So next, which is evolved from this, is what this, that embryo mass cells are arranged in 3 layers, that is ectoderm, mesoderm and endoderm, hence triploblastic. But see here in mesoderm there is a clear cavity, so that organ and organ systems are developed and can easily arrange in that cavity. So see, therefore this embryo is called triploblastic coelomate. I hope you got it. Same embryo mass arranged in that evolutionary trend into these 4 types of embryos: triploblastic coelomate, triploblastic pseudocoelomate and triploblastic coelomate. Here in this evolutionary trend, see the miracle that diploblastic coelomate in porifera, they have only cellular level of organization, but evolve into tissue level organization in coelenterate and ctenophore. So all these 3, they have diploblastic coelomate. But next level, platyhelminthes which evolve, they are triploblastic coelomate. Because of presence of mesoderm, they started to develop the organs, but no organ system. In this case, with pseudocoelomate, they begin to develop organ system, although it is poorly developed, but begins to develop the organ system because they got mesoderm as well as cavity to adjust and accommodate that organ system which are developed in it. And in third case, because of clear cavity with mesoderm, they are evolved to have all kind of organ system, that is animates onward up to human being. They are triploblastic coelomate with all types of organ system, and that is what in the evolutionary trend from the same blastula these are evolved, and therefore these are the group of organisms are classified according to this embryo stages, and therefore very first or important character as an event supposed to be write general characteristic of phylum, in that this will be the important characteristic. Suppose a porifera you have to explain, no doubt you write another character, but you will be knowing that what is diploblastic coelomate and why porifera are diploblastic coelomate, and hence onward that same you can use the characteristic. I hope you did understand how these embryo mass are arranged into this 2 layer, 3 layer without coelom, 3 layer with pseudocoelom and 3 layer with true coelom. So this was the concept that is supposed to be clear. I hope you did understand. This is a short video. You can enjoy my previous video which regarding development of embryo stages, but I thought of to make it clear, therefore I have drawn separately so that you can understand from this common structure this also develop, this also develop, this also develop, this also develop. And hence in the evolutionary sequence I arranged according to this. This is the previous one and primitive one, and this is the advanced one, and that is what clear to you. I hope you did understand. Even if you are having any doubt, you can ask in comment section. I appeal you to like and share this video to your friend, and if you are new, please subscribe it and enjoy learning.